The overall goal of the following experiment is to develop a two-dimensionally patterned substrate for the control of protein adsorption and cell growth. This is achieved by using photolithography to fabricate a patterned master, which is used to produce a PDMS stamp for microcontact printing. As a second step, the patterned gold substrate is prepared, which involves microcontact printing to produce a pattern with regions that absorb and resist protein. Next, protein and cells are added to the substrate in order to visualize the pattern and study cellular growth and behavior. Results are obtained that show good protein and cell confinement on these patterned substrates based on fluorescence and phase contrast microscopy.
The main advantage of this technique over other existing methods, such as microcontact printing of proteins, is that the glycol-terminated monolayer resists nonspecific protein and cell adsorption in the background of the pattern.
Generally, individuals new to this method will struggle because proper stamping technique must be optimized for each pattern printed.
To begin preparation of the patterned master, center the silicon wafer on the spin coater, and during the initial step of the 2-cycle spin program, rinse the wafer with acetone. During the second step of the spin program, the acetone will evaporate, leaving a clean, dry wafer. At the beginning of the spin, apply photoresist to the wafer, and then spin coat using the conditions described previously. Soft bake the photoresist-coated wafer at 110 degrees Celsius for 2 minutes using a high uniformity hot plate. Next, photo-pattern the photoresist-coated wafer using a mask aligner system and an appropriate mask. First, place the substrate onto the vacuum chuck of the mask aligner. Then insert the mask into the holder tray and turn on the vacuum to hold the mask in place. To confirm good contact between the mask and substrate,